Sure. So we're interested in serious psychiatric disorders, and we're interested actually in the role of inflammation and whether by blocking inflammation you may be able to prevent some of the events that occur with these disorders. So the main disease I talked about was mania, and mania is a common problem around the world. People with mania in between events are usually pretty well fit in society. Many of them are doctors and teachers and workers and students. But when they have a mania episode, their mind isn't working correctly, and they may think that they can do things that they can't, like fly, or like interrupt meetings, or like walk into traffic. So people with mania, most of the time are doing okay, but when they have the event, it's quite dangerous for them. And what we've been able to show is that people, when they have these mania events, often have evidence of inflammation. And we've also been able to show that sometimes this inflammation is stimulated by some things that they might eat. And we've also been able to show in a controlled trial that feeding a probiotic preparation to people that already were hospitalized with mania was able to prevent a subsequent episode within a 6-month period of time. So we're very interested in whether we can identify people with mania or perhaps other psychiatric disorders as well, and whether by interrupting this chain of inflammation, whether we can try to prevent further events.
Excellent. So I understood from your presentation that basically your initial work was based on a cohort, or what you call the Shepherd-Pratt Exposure Survey. Can you maybe...
Yeah, so this is a cohort of individuals that come to Shepherd-Pratt Hospital, which is a private, nonprofit hospital in Baltimore, and they take care of all sorts of patients there, patients with a range of different disorders. And many of the patients that come there are enrolled in a clinical trial. They're asked to be a part of a trial of one form or another, and we also enroll healthy controls who we put advertisements in and say, do you want to be part of a trial? And the study has gone on for about 15 years, and we've enrolled more than 2,000 individuals. We have blood samples, and we follow them. So we've learned a lot from this cohort, both from the problems they have when they come in, or what happens to them over time, because we follow them over time. And the people with mania that I talked about are part of this cohort that we've been able to follow.
And so basically you mentioned that there was really a difference in terms of diet for the people that were exposed to nitrated meat in particular?
Yeah, so we found, we asked a lot of questions of these people, and we asked a lot of questions about their diet, what types of food they were eating, when they were eating it, and so on. And we found, somewhat to our surprise, that the one food that was most associated with a problem, and particularly with mania, was eating cured or nitrated meat products. This would be beef jerky, turkey jerky, meat sticks, and things like that. And this was very surprising to us because it was not something that we had considered before. But when we looked into detail, we found that these products contain nitrate. And when we tried to look at an animal model of the effect of nitrate, we were in fact able to show that feeding these products to animals also changed their behavior in a way not exactly like mania, but was somewhat similar, and also changed the expression of a lot of factors in the brain that are associated with neurotransmission. So we think it's the nitrates that are probably causing problems. Both people that were taking meats without nitrates, typical meat preparation, or the animals, the rats that we gave meat preparations without nitrates, didn't have any problems or any differences. So we think it's the nitrate that was causing the trouble.
You have also further explored the possible mechanism of action by using models with antibiotics?
Well, it's a similar set of studies. We're asking the question of whether changes in the microbiome might be associated with problems. And we found in this cohort that one of the things that people with mania had reported having done in the period before their hospitalization was, in fact, that they had been given antibiotics by their doctors. And in fact, it was also true of some other psychiatric disorders as well, but most typical of mania. And it was a range of antibiotics, but it was typically antibiotics that would have a broad spectrum. That is, they could deal with a lot of different bacteria. So from this study, we postulated that antibiotics were causing problems in people who were probably predisposed to mania, but then were tipped over by a change in their microbiome. And subsequent to that, we've looked at large populations, that is not patients in a hospital, but it's individuals in a population who were involved in a large healthcare system. A particular study we looked at were people involved in the healthcare system in Denmark, in collaboration with many fine doctors and researchers from Denmark as well. And we found in that cohort that individuals who had been exposed to antibiotics early in life had a range of psychiatric problems later in life. And it wasn't just mania, it was a range range of disorders, although many in fact were mood disorders. So this was a different type of study. It was a cohort study, but I think it showed very similar results. And the best way we can interpret this is that if you change the microbiome, at least in some people, that's going to cause changes in mood and behavior.
But you have seen some kind of a specific effect of specific classes of antibiotics, right? Not all antibiotics, it's specific categories, or?
It was not, yes, it was not all antibiotics. It was not just one antibiotic, though. What it seemed to be was the more broad spectrum the antibiotic. That is, the more different bacteria that the antibiotic would hit, if you will, or kill, the more the risk was. So there's some antibiotics that only are effective against one type of bacteria. So, for example, macrolide antibiotics, such as erythromycin, have a relatively narrow spectrum, largely gram-positive organisms, and they didn't have as much of a problem as some of the more broad spectrum antibiotics. So I think that has some practical indications, because we're not telling doctors not to use antibiotics if it's necessary, but we are saying a doctor should prescribe antibiotics that are most appropriate for the infection that they're trying to treat and be as narrow spectrum as possible. And this is quite consistent, I think, with public health advice to try to minimize the development of resistance. So it's for a different reason, but it's the same recommendation. So we're quite interested in prevention. So that would be a way of putting it. It obviously would be wonderful to treat everybody that has psychiatric symptoms, and I think that's the ultimate goal. But I think where we are now, the best we can do is try to prevent episodes from happening. So in order to do that, we want to identify people at high risk and then provide an intervention. So people with mania, I think, is one example of a high-risk group. Someone that's been hospitalized for mania is very likely to have another episode of mania. There are other high-risk groups as well. Someone with a suicide attempt, for example, regardless of what their diagnosis is somewhat more likely in the general population to have another suicide attempt. Also probably true with someone with a severe episode of depression. So at least our short-term goal is to try to identify these people and then provide an intervention where we might be able to help them. The long-term goal, of course, is once we understand more about what's going on, would be to treat everybody that has these disorders. But I think that's more in the future than, I think, trying to prevent. And the principle is that prevention is often easier to accomplish than treatment. It's often easier to prevent an infection or prevent a disease than it is to treat it, and that's where we are now.
Just wanted to know, what were the challenges you faced when conducting such a study?
Well, the main challenge is funding. We've actually been fortunate because a private foundation has funded us, but I think these studies are expensive. To do them right, the patients require a lot of care, a lot of checking, and so on. So that's probably the major problem we've had. Fortunately, the other problem usually is compliance. That is, will the patients stay in the study? But fortunately, at least the probiotic study that we did, the patients seemed to tolerate it quite well, and the patients usually did a good job of staying in the study and taking the medications. So I think it's usually just the matter of funding and doing a study that's large enough. That's probably the main issue.
And so what's next for you and your team, I mean?
So we'd like to do more trials. So this was a small study in mania. We'd like to do first of all, some additional studies in mania. We used one type of probiotic. There are many others that might be used, so those could be done as well. We're also interested in some other disorders. We're interested in bipolar depression. So those are people also with bipolar disorder, but their main thing that brings them into the hospital is not mania, not overactivity, but severe depression. So that's another problem that people with bipolar disorder have. And we postulate that the same mechanism will be involved and that it might work. Probiotics might work, but we actually don't know that. So those are the main goals of what we're trying to do now.
Okay, and so what would be your key takeaway message from all the presentations that you've listened to over the past two days?
I think this is a very promising area. I think that it's a very early area. I think it needs a lot of support. It needs good, careful clinical trials. It needs good animal studies, and it needs good support, financial support, I'd say, both from governments and also from private companies and foundations.
We run a clinical trial in my country, and we have a look through the effect of the probiotic and prebiotic on psychological outcome in depression patient. Well, as you said, the question about the previous study in this field, there is little information in this field of the depression people. Only 3 studies have been done so far, and they show that 2 of them they showed as some strain of the probiotic can improve the symptom of the depression, and our study confirmed this result and show that we can use of the probiotic to improve the sense, the mood, even the stress of the patient, with the patient who's suffering from depression.
How did you select the specific strains that you use in your study? How did you came up
Well,
with
you're
that?
talking about the strain, I think is a difficult part of the study, because when you have a look through the different strain being used in the previous study, and we found that the Lactobacillus helveticus and Bifidobacterium longum are the most maybe effective strain than in depression patient, or even in the animal study. So that was the reason we choose this one. Actually, we use the Galactolactobacillus. We use the GAS, which is the prebiotic, and then we found that in the animal and human study, this one can grow the Lactobacillus and Bifidobacter. So with these two intervention, we have a look through the effect of this one in the depression patient, and we found that the prebiotic maybe is less effective, but the probiotic definitely have some effect on the improvement of the symptom. Well, the result of our study showed that the BDI, which is a scale for measuring the depression symptom, will be improved when you give them these two strain of the probiotic. Also, we showed that in clinically we can decrease the stress level, which measured by cortisol, and we found that we can decrease 20% of the cortisol after administration of these two strain of probiotic. And actually, if we say it in scientific word, the ratio of the tryptophan to kynurenine, which is a marker of the produce of serotonin in the mind, will be improved by giving this two strain of probiotics. So that was an interesting result of our projects. Actually, we are going to have a look through the data by measuring the BDNF, which is an interesting area for the neurotransmitter. In future, we are going to publish this one, and also effect of the probiotic on appetite. You know, in some of the melancholic patients, they losing the weight, they have some lose of appetite. Our study showed that if you give them the Lactobacillus helveticus and Bifidobacter longum, we can improve the appetite of the patient as well. So this is another interesting result. Also, we have a look through the effect of the probiotic on immune deficient patient, and we found that this is quite interesting area to work on, and can improve the CD4, which is a marker in the these diseases, and you can improve the CD4 by using the probiotic.
What's your own expertise? From which expertise do you come, and where?
Well, I'm the nutritionist actually in the clinical nutrition. I'm working in the Tehran University of Medical Sciences. I'm in the Department of Clinical Nutrition. We run plenty of clinical trial to show that what's the effect of probiotic on health, and you know that there is plenty of basic science here, but when it's making difference, that you can try it in the clinical field, and I'm happy to run a couple of studies like that in my country.
Yeah. I'm curious to hear what you think of Mind with Microbes as a conference. Is there something you like about it or don't like about it? How did you experience this 2 days?
You know that I like this field, you know, and I am sure that this is not a new field. Before the history of civilization, the people facing with microbiome, with the bacteria, and this is another, I think it's not a new field, it's a very, very old-fashioned field. And I'm really, really happy to work in this field. And I know that with this environment, exposure and with a different kind of antibiotic, maybe the solution for improving the gut microbiota, using of different fermented food and or maybe supplement to improve the composition of the gut microbiota. And definitely this is a very interesting area to work on it, and I'm happy to be here in the Netherlands. This is the first time I came to the Netherlands. I'm really, really happy to see this beautiful city, and hopefully I can come back again in the next future. Yeah.
Yeah, so my talk today was about the microbiome and how it interacts with diet to influence neurological disease. So specifically, a key message was that the microbiome seems to be important in mediating the effects of the ketogenic diet, in our case on seizures. So I drew upon existing human research on this diet, and so the ketogenic diet is already used clinically to treat refractory epilepsy, primarily in kids. Our new work is really in animal models. We also used existing animal models for epilepsy, but we studied how the microbiome changes in response to the diet, and we also studied whether the microbiome is important for the effects of the diet. And so our findings were that the microbiome is both necessary and sufficient for the anti-seizure effects of the diet. And we studied a little bit also about particular microbes, particular species that seem to mediate it, and particular activities from these bacteria that are important.
Yeah, I found very fascinating basically that you demonstrated that you really needed the two type of bacteria that you have identified to have them together rather than it was not working basically when using only one of the phyla or species.
Yeah, that was actually an interesting surprise from the work in finding that we needed kind of a community, a pair of bacteria together to mediate the anti-seizure effects. And so that's really interesting on a basic science level in that our evidence was suggesting that the bacteria kind of speak to each other, that there's crosstalk through chemical signaling between the bacteria, so that that kind of interaction is really important for the outcome.
And you mentioned also that actually you've tried things with heat-killed bacteria and that basically it was not working. Did I got that right? So
yeah, there were actually adverse effects with dead bacteria, heat-killed bacteria. And so this is also consistent with the rest of our work, where it seems like the bacteria have to be alive and actively functional. So actively interacting with dietary components and metabolizing it to mediate the response.
So from this work, what would be the next step for you or your team?
Yeah, there's still a lot to be done on the basic science side, particularly in studying neural circuits that are effective. Most of the work that I spoke about was periphery, so characterizing the microbiome, characterizing microbial functions, maybe a bit on blood metabolites, and we barely just talked about neurotransmitters in the hippocampus. And of course, it's not just neurotransmitter levels, it's how they work within a network, a neural network. And so more basic science on the neuroscience would be useful. Aside from that, another area that we're trying to expand in is on the clinical translation side. So we can do all of this really, fun basic science, but at the end of the day, we don't know whether it's going to be useful for people. And so we're really trying to start a human study to see whether our findings in animals is relevant at all to refractory epilepsy. So what I really enjoyed about this event was the mix of expertise in the community of attendees and even the speakers. Since I'm not a clinician myself, I really enjoyed a lot of the clinical talks on neuropsychiatric disorders and upcoming talks I'm sure I'll enjoy on neurodegenerative disorders. And it's really interesting to see, how much more there is to be done on the clinical side. I was really humbled by today's speakers when they were addressing, both the promise of the field, but also results with existing probiotics, and that many of the probiotic studies done to date in humans have not been that effective. And so I think that's an opportunity for us to really work hard at, trying to make what we find on the basic science side translational. I mean, I love the entire field. So actually, I spoke about epilepsy today, but my lab has very diverse interests. One really interesting area is in neurodevelopment. So we had a neurodevelopment session yesterday that I was really drawn to. And just the general idea that, during development is when, animals are most reliant on sensory signals from the mother and early life signals from the environment. And so maybe that's where the microbiome can really, elicit a very impactful role. Yeah, aside from development, I'm also interested on the other side of the lifespan and neurodegenerative disease. I think that's increasingly a really huge public health burden is a neurodegenerative disease, aside from being very devastating for families. And so it's intriguing to see the links to microbiome arise. And that's another area we're really interested in contributing to.
Oh, great. So just maybe as a final question, what do you think about the event? Was it your first time? No.
No, so I was here at the first
edition,
one.
So maybe you can see some changes in the content, in the people.
Yeah. I mean, the science is always advancing, so I find that the most exciting part. It's been a great community of people. I particularly enjoy, that the presentations where speakers feel comfortable presenting some unpublished data and really get into the rigor of the work that they're doing. I think that can't be said for all meetings. There are other meetings where you don't really have that type of community to be able to share. The poster sessions have been also really fantastic, really, really exciting upcoming work. So I'm glad to see that there have been a lot of, faculty members and also trainees, postdocs, and students here to present their ongoing work.
So what we found is that the gut microbiota can modulate the immune response, so change the T-cell response specifically towards either a pro or anti-inflammatory response. And since stroke has a strong inflammation, there is strong inflammation after stroke, modulation of this immune response by the gut microbiota can influence stroke outcome. So this was the link. So T cells, a very important mediator between the gut microbiota and stroke. So I'm working on the gut microbiota in stroke, and we have found a direct link of the gut microbiota, how it can influence a stroke outcome, and this is via regulation of the immune system. And this has been done in a rodent so far, and we don't have yet data on humans, so stroke patients. So we have some preliminary data. So the idea is how we can modulate the gut microbiota and influence stroke outcome.
Yeah.
So we are working on gut metabolites because we can, for instance, modify the gut metabolites through the diet. And we have some preliminary data on stroke patients showing that actually the tryptophan metabolism is also affected in stroke patients. So maybe we can also relate our findings in a rodent to patient. So what we want to do, so we're gonna launch this year, is like a study of feces from stroke patients. So we're gonna collect feces and follow stroke patients and actually analyze so all the bacteria that are in the feces and also metabolites.
And are you going to intervene as well to modulate the gut microbiota?
Yeah, so ideally it's what we want to do. So today we heard about probiotics, prebiotics, and that can influence other diseases, so why not in stroke as well?
Okay. So and what do you think are the most promising directions considering gut-brain?
Yeah, I think there are still a lot to do, and I think still the gut is a black box and very complicated, and especially we don't know enough about mechanistic but it's good we are still doing very great research and progress. It's a very young field, so I guess in the 10-20 more years we will know much more.
What do you think of the conference, by the way?
Yeah, I think it's great. What I liked a lot also, it's so different people from different fields, different disease, and studying the same gut-brain axis. So also it gives new ideas on how to tackle these questions. and also, of course, doing networking and maybe collaboration.
Yeah.
So I very much like the talk before me about seizure and the effect of this bacteria, and the way Il and Xiao addressed the question, so tested actually this bacteria and found an impact. So yeah, this kind of methods, I would say maybe it's more experimental, but this is very helpful.
Yeah.